Prue Francis and Paul Carnell. I'll give you a little bit more information about them in a second. So this is probably a good place here to acknowledge the traditional owners of the work that of the lands on the work that we've been doing. So we're in Victoria, right down the bottom of the mainland Australia in Victoria here. So our two sites are these little dots that you'll get to be familiar with during the talk. This is on Bunurong country, and today I'm on Wadawurrung country for anyone that might be coming from a similar place. And I want to acknowledge the traditional owners here and everywhere that everyone's coming from, and their elders past, present and emerging. So the issue we've got in Victoria for many of our attendees today, it's not going to be new information, that urchin barrens have become a problem in this part of the world and have led to loss of kelp. So on the left-hand side, beautiful kelp area, and on the right-hand side, moving to barrens. So it's thought at the moment, estimates there are about 130 million of the native urchin, Heliocidaris erthryogramma, in the Port Phillip Bay area. I think we've got somebody maybe in the background that's not on mute. Thank you very much. So we've had a lot of biodiversity loss with the loss of this kelp, and the urchin abundance in this part of the world at least has been related more to water quality and drought interaction rather than climate change per se. But after the millennium drought, we had a really huge increase in urchins that then didn't have a lot to feed on, so then they started to feed on the kelp. So we've got these losses. So our restoration approach to try and improve this is this host of different people. So Parks Victoria looked after urchin management. Ourselves and the Nature Conservancy looked after kelp cultivation and outplanting, and the University of Melbourne have been doing monitoring at a larger level across two sites. So here's the big project team. So the kelp cultivation and outplant trials were led by all these lovely people at Deakin University, and also our collaborator Kane Layton at the bottom from Blue Conservation Services, who was teaching us how to cultivate Ecklonia radiata. So Dr. Paul Carnell, myself, Dr. Prue Francis, Jasmine Bursick and Kayla Gorn has did a honours project that interacted with this work, looking at light intensity and temperature that we could use for the different cultivation parts. And oh, God, goodness me, maybe I won't say everybody's names out loud because otherwise we'll be here a lot, but here's the lovely faces of all the different people involved, and the co-authors on this, this paper are our cultivation people here and Scott Breshton, who did a lot of the diving and logistics and organising all our getting them out into the field. So to cultivate Ecklonia radiata, we first had to go out and collect it from populations where it was healthy. So we go out and snorkel, and this site's called St Leonard's, and we go look for adult tissue. So when it's reproductive, it starts to, you get a blade, or you can get the middle part, and you go and collect it. Take it back to the back of the car, have a bit of a look under the thing, and this is like a perfect textbook one, the one in the middle of what we're looking for, to look for the sori. We want to get ones that aren't too covered in epiphytes and those sorts of things, so as clean as possible. We give it a trim up and keep it on ice and take it back to the lab. When we get back there, we give it a really good clean in iodine, diluted iodine, and we make sure that we've got nice dry pieces. They get a nice dry, and then they go sit out for a while, and so they start to, it actually induces them to release their spores. Once we've got our spore solution, we check how much we've got, and we choose a density that we want to do, which we've done from experiments. And then we seed it onto green gravel. So this is our research technician Jasmine seeding out of a beaker into the laminar flow onto our green gravel, which are basically just these big bits of gravel rock in a tray. And then they start to grow. We put them in the aquarium under set light and temperature, and we give them a feed and a water change, and they grow for about 6 to 8 weeks before they're ready to go out in the field. So this is an example of what they look like. So this is on twine, which is often used for aquaculture, and this is on the green gravel. So if you look really closely, they don't look like much on there. They look sort of like moss or something, but up close, they look quite beautiful, little tiny baby sporophytes. So this is the TNC crew. When we got to go to the outplanting part, they went and did all the diving, and they put these plots in. So they did a lot of work putting those out. This is them carefully putting out the twine. So there's some rebar into the rock here, And all the little babies are on the twine there and they're wrapping them out. So here's some more information about the design. We've got these two sites, Jawbone up the very top north, and to the northeast we've got Ricketts, and these are both actually marine sanctuaries. And in these places we've got permits to cull the urchins, so they're native. We didn't get rid of all of them, it was just reducing the density. So some of the densities could be extremely high, like 60 per metre squared or more, and getting them down to 2 per metre square, which has been shown to be sort of the natural limit of where they stop creating barrens, and they're just part of the ecosystem and not eating all the kelp. So in this trial we had green gravel and twine that was 12 weeks old when we planted it. Here's a little bit more about the design. These are the two sites. This is what it looked like if you wanted to have a look at the field. In Jawbone, we've got these twine that's tied around rebar that was recessed into the rock. And the green gravel here, you've got little tiny bits of gravel you can see with their little fuzzy kelps. And on the right-hand side, we hadn't put the rebar in, so we used these Besser blocks to tie it and attach it down and marked it so we could go back and see its success. And we had 7 replicates of everything, and we monitored them 2 weeks after and 2 months after and since, but I've got the data today from the 2 weeks and the 2 month time. So we went out to monitor it. We did it on scuba. Here's Paul skippering and checking us all out on the research vessel. You can see it's really quite close to shore. It's less than a kilometre away, probably only about 400 or 500 metres. And this is the bit you're probably all dying to see, is how we went. So we used a mixed provenance culture. So we collected tissue from St Leonard's and also from Williamstown, and this was adjacent to the site at Jawbone. And we mixed their spores together in the lab, and they seemed to grow quite well. So we use those for the restoration because they seem to do the best. Up the top, we've got all the gravel and how it's grown from the time it was planted out to 2 months. And at the bottom, we've got twine from when we planted it out to 2 months. So it might be kind of obvious at first glance that the gravel did a little bit better, but it also started out a lot bigger. So it's caught up quite well. And even though it looked like there wasn't too much improvement happening at the 2 week mark, we sort of did photoquadrats, just like little small photoquadrats within each plot. And it was like, oh, there's only this teeny tiny little kelp, you can barely even tell it's a kelp. But then by the 2 month period on there, you got lots of them all on top of each other to 4 centimetres. So some of them might have been too tiny to pick up at the 2 week mark, which was quite challenging to try and find them. Sometimes they were hiding from us. But yeah, we had 100% success on all the gravel that went out at Jawbone, and we started to get, I think we only got about 70% success on the twine, so not quite as high, but pretty good. And at Ricketts Point, the other site, you get an extra bonus one here because T&C did some other monitoring and happened to get some lovely photos between our monitoring. But similar story, we started to get some really big improvements at the top, and we've even got a little tiny fish. I got surprised twice doing monitoring by weed fish in our kelp, trying to measure the length. And this little dude gave me a surprise, and so he was extremely cute. And it's so nice to just see that we're not just getting kelp regrowth, but so quickly starting to get things going and living in it was pretty exciting. Yeah, so even very similar story to the one above, getting to about 7 centimetres after 2 months, and the twine getting to about 2 to 3 centimetres. But then even just 1 month after that, the amount of growth that you can see in the final panel of 17 centimetres. So that grew crazy in the top part. And so, but these were planted out at the very start of summer. So we think that was a really good time to plant them because they were growing quite well. But we did lose a bit of twine at Ricketts Point, which was a shame. We're not sure if anyone bumped it or whether it just broke. So that was also something that we had to manage. And we also lose some gravel. Some of it rolls away or disappears, and some of it's just grows in with the substrate, so you can't actually distinguish it from the rock itself because you start to get lots of turf algae and other things growing back. So there's a bit of interaction between the urchins that are being culled and also the planting, and that's some of the work that Melbourne Uni and T&C were doing with looking at the design over a much larger scale to look at just areas that were culled versus the ones that were planted as well. So in summary, it's short and sweet, The success happened on gravel and twine. The gravel was a bit higher in success and growth at that period at least. And at 6 months, we haven't been able to present the data to Gage, and we're still doing some collections. Still showing some success, mostly on the gravel, but we've also got some improvement in the twine that seems to be growing as well. So both are viable options. It might depend on your application and the site that you've got. And yeah, we the whole team did a great big upscale, so there was an awful lot of kelp growing at the lab, so up to 1/2 hectare. So there'll be more information coming into the future. Yeah, we have loads of people that helped in the field, and here's some silly pictures of us out in the lab in the field doing all the work and all the different people behind the scenes that helped with skippering and lab and all that sort of thing. And this was funded by the Victorian state government. But yeah, that's it. Thank you.
Lovely. Thanks so much, Jackie. I'll ask kind of a general question to kick us started, what people think of theirs. And then afterwards, if you have a question, just raise your hand and we'll call on you to unmute yourself and ask, or you can ask in the chat as well if you don't want to use the microphone. But Jackie, given the 2 different methods had good success, were there any sort of differentiating factors or conditions that you think might make them more appropriate given certain environmental scenarios, or when you might prefer one over the other?
Yeah, it was actually, yeah, absolutely. Yeah, it was interesting because our collaborator Kane has done a lot of work in Macrocystis in Tasmania, and they found a lot more success with the twine, but it was much higher wave energy areas. So they were surprised to see our results actually kind of went the other way. But these areas, particularly that part of Port Phillip Bay, is really quite sheltered. So there's low water movement compared to like an open coast site. So that could be why we're getting really good success with the gravel. Yeah, I guess the twine definitely probably stays in place a bit easier if you didn't have the right size gravel. If you use something that's too small and it sort of floats away. I think some other studies have found that if they had too small gravel, once it started to grow, it created a sail and disappeared. But then if you're having it too big, it's quite hard to move, and you also want it to be able to grow onto the natural substrate and just not be an artificial reef. So there's a few things in mind. Ours were about 20 centimetre sort of size rocks. The first lot we lost a few, especially on the flat bits where there was sort of a drop off a boulder. You'd find them down the side of the boulder, but then sometimes down there they were like, here's good, we can grow here, and you'd find one. It became a bit of a finding mission when you're trying to find the rocks. But then you're like, it's gone over there, but it's happy. So we'll leave it be. Just made monitoring a challenge.
We'll find one in Sydney. It'll take off on a sail trip.
Yeah.
We have time for one question for Jackie, if anyone has anything they'd like to ask. Yeah, Max, go ahead.
Hi, thank you for the presentation. I'm curious about the urchin calling. If you could talk more about that and explain the methodologies and kind of what density you have and how successful
they may just land as an ambassador rather.
Sorry, Max, I don't know about for others, but it went kind of funny, echoey sounding for me. Do you want to type it in the chat?
Sure, yeah. The chat is disabled for me, so
that's better.
you can check.
You're not echoing.
Is it better?
Go ahead.
Okay, yeah, I was going to ask about the urchin calling, and if you could talk more about what methodology you used, what densities you're aiming for, and what you see kind of year on year if the urchins start coming back, and how you would deal with that.
Yeah. So they got contractors. So commercial divers to go out and do the culling, and they were reducing densities down to about 2 per metre squared. So if you're doing that in practice, often because there'll be some hidden away, you can pretty much knock most of the ones that you see on the head, I guess. And they've been doing when we do monitoring, if we start to see that the densities come back, they can roll out a second culling. So that's going to be sort of the longer term issue, is how much we start to get back, particularly because we're only culling in these sections. If we culled the bay, it would be very, very expensive effort. So there's been lots of work being done by a range of people trying to figure out how we manage the urchins on a larger scale with fisheries and collections and that sort of stuff, as well as culling, because it is a commercial species and a native species, so it can be a bit challenging. But yeah, 2 per metre squared was the density we got down to, which is based on a few papers that have come out prior. I can find them to share with Aaron, to share with everyone. If that helps.
Yeah, happy to send around. And I think we've seen sort of numbers between 0.5 and 2 meters squared for densities that sort of have that threshold. There is one more question in the chat, but just keeping on time, Jackie, I might ask you to stop sharing and we'll pass it over to Madeline to talk to us about thermal tolerance, the big question of the day.
Thank you. Go ahead and see. Okay. Okay, can everyone see that okay? Okay, thank you. Great, fantastic. Hi, my name is Madeline Harden. I am a recent graduate of the Nugstein Lab at the University of Southern California in Los Angeles. And today I'm going to be talking about some of the work that we've undertaken collaboratively over the last 6 years, in which we sought to elucidate the genetic underpinnings of giant kelp heat stress response in Southern California. So to begin, I want to introduce our species. So we're working with giant kelp, or Macrocystis pyrifera, and those native populations that exist along the Southern California bight that we sampled, that eventually became what is now the Alberto Nugstein germplasm collection. And then and when I say germplasm collection, I mean kelp seed bank. And how we then use that seed bank in collaboration with Dr. Rusty Lansford's lab at the Keck School of Medicine to develop a heat stress phenotyping screen for our gametophytes. And those gametophytes, of course, were a subset of the germplasm collection. We used an imaging system known as IVIS for our phenotyping screen. And IVIS is a non-invasive imaging technique. It's used to visualize living organisms, and it tracks the biological processes, oftentimes leveraging either fluorescence or bioluminescence. In our case, what we're leveraging is kelp chlorophyll's ability to autofluoresce, and we use that as a proxy for overall cell health. So in this part 1, I'm also going to discuss a correlation analysis. So we were able to score phenotypes for heat stress in our gametophyte life cycle stage. But we were also very lucky in that we had a data set from 2019 in which genetically similar kelp sporophytes, so the juvenile sporophytes, were outgrown on an open ocean farm in Northern California under warmer than normal ocean conditions. So Maxim Kovalev, Sergei Nugstein, and others in our lab ran a correlation analysis so that we could see if the heat stress tolerant phenotypes that we were seeing as gametophytes could actually be seen at that adult sporophyte life cycle stage. And we actually published this data in August of this year in Communications Biology. And then moving forward, I have a part 2 of my talk. This is unpublished, but we used the results of our phenotyping screen for Macrocystis gametophytes to then run what's known as a genome-wide association study. And the purpose of this study is to understand if there are proteins that might play a regulatory role in how giant kelp respond to heat stress. And then for my conclusion, I'm going to be talking a little bit about some of the kelp restoration initiatives that are happening right now in California, as well as next steps for this research. Okay, so just to begin, this was the impetus for my work. So as I know I'm speaking to an audience of kelp enthusiasts, so it might come as a surprise to no one, but kelp forests globally are under threat of global climate change. And there's this amazing amount of information that's been coming out. This year alone, we have so many brilliant papers that have been published that show us the detrimental impacts that global climate change, namely sea surface temperature increases, decrease in nutrients, increased ocean acidity, all of these things, all of these abiotic stressors are negatively impacting the physiology and overall reproductive success of kelp gametophytes and their adult sporophyte counterparts. We see a decline in photosynthetic capacity, growth. Oh, sorry, nope, okay. And as Jackie was actually just talking about, we also see this increase in herbivory in Southern California, excuse me, in California, not in Southern California, but in Northern California. Dr. McPherson and her team published a paper which was incredibly stark. It shows us that over 95% of the Northern California kelp canopy got lost following a major warming event in 2015. We had a massive drought year, and then we also had what was known as the blob in the ocean. So it's just this mass of warm water and extremely high salinity. So Dr. McPherson used satellite telemetry in this case. So again, leveraging the chlorophyll to show us just the total and utter collapse. And then as in Australia, we have a proliferation of purple sea urchins. So we have urchin barrens as well, which is a massive problem. Luckily for us as scientists, we can leverage the kelp biphasic life cycle, right? So on the left-hand side here is our giant kelp. What's not shown below is the reproductive blades known as the sporophyll tissue. And actually, Jackie did a fantastic job of already talking about how one does a sporophyll release in the lab, where you excise that reproductive tissue, which has the ripe sorus in it. You take it into a lab in artificial seawater conditions, and you do a spore release. And then luckily for us, actually, you know what? Can people see where I'm pointing at with my pointer? Okay, fantastic.
Yep, you're good.
Great. Thank you. Okay, so yeah, so we have the zoospore release, and after a couple of days for giant kelp, you can start to see the formation of the sexually dimorphic male and female gametophytes. So this is a bright field view of our female and male gametophytes, and I just kind of want folks to focus for the first part up here in this right-hand corner. This is an actual image of our germplasm collection that we have in our lab. We have our kelp gametophytes in these cell culture tubes, and they're separated by sex. And what we can do is we can arrest them in that gametophyte haploid life cycle stage indefinitely at specific light, temperature, and nutrient conditions. All right, so going back to our populations, in 2015, Johansson et al. Discovered via microsatellite data that these populations along the entire Southern California bight here are genetically distinct. So we have our northernmost population in Arroyo Quemada, which is essentially Santa Barbara, all the way down to Camp Pendleton, which is in essence San Diego. It's close to the Mexican border. So there's a little bit of a 1 degree temperature gradient on average from north to south here. So these four populations, in 2018, source tissue was collected from each of these populations. And at the Alberta lab, over 500 individuals were then sequenced and then made into our germplasm collection here that we have now housed in the Neustein lab as well. So that's a little bit of background on our germplasm collection and the biomass that I used to develop our phenotyping screen. So just to kind of provide a little bit of a workflow for what we did in Rusty's lab at Children's Hospital, we started with our sequenced genotypes. I used 204 and I put them in replicates of 3, randomly plated across 96 wall plates. They were treated in these benchtop incubators. The only variable I changed was the temperature. I didn't adjust. They had the same nutrient, light, everything else. The only thing that was changed was the temperature. Then I used the IVIS intervital imaging system. This is an actual image that's output by the IVIS. So what we're seeing here is our 96 well plate, and in each one of these wells, which is like a little tiny indentation that you can fill with seawater and your kelp gametophyte, the red and yellow pinpricks are actually your whole kelp gametophytes fluorescing. And this data that's collected is actually a discrete variable. So what it's called is a fluorescent intensity variable that gets output, and we can use that as our actual phenotype as a proxy for the cell health. At the same time that I was doing this, I wanted to see what the actual organelles were doing. So I would take one of my samples, a single kelp gametophyte, and I would take just a little bit of it. So I had multiple cells that are clonal. I would fix them and stain them, and then in formaldehyde and dye, and then I would image them using something called confocal imaging, which allows me to see the nuclei, the cell wall, microbiome, and the chloroplasts. So I have that data as well in tandem with the IVIS data. And then once I had collected all that information, I went ahead and we ran something called a genome-wide association study. So for a little bit more background, I screened approximately an equal number of individuals per population, approximately 50 per each one of those 4 populations along the southern coast of California. For my temperature treatments, I used a control of 13 degrees Celsius. Then I used 4 heat stress treatments of 21, 23, 25, and 27. And I used a time frame of 4 weeks for heat stress, and then I did 4 weeks of recovery. And I'll just say right now that the 4 weeks of heat stress are the only thing that matters. There was actually no recovery phenotype. So we didn't actually see any of the kelp gametophytes recover. So what we did was, in the beginning, we would take all of our well plates and we'd put them in control treatment and then the subsequent heat stress treatment for 4 weeks. We'd collect data at day 0, day 14, and day 28. And then once day 28 happened and we'd collected all of our data, we'd immediately put all of these well plates back into the control treatment and leave them for 4 weeks and collect data in the same manner. And for that component, for the recovery component, we saw no change, no recovery. Okay, but what we did see for the heat stress treatment was actually really illuminating. So we collected, again, our fluorescent intensity data, and visually this is what this looks like. So there was no change from 13, 21, and 23. They were all the same. So at 21 and 23 degrees Celsius, the gametophytes were happy and healthy. However, if you'll notice here, at temperature 25, so here we have week 0, these are happy, healthy gametophytes. They're robust, they haven't been heat stressed at all. At week 2 for 25, you can kind of begin to see there's a shift, there's a change in the fluorescent intensity just visually. And then for 25C. And then at 27, you can really start to see an extremely stark difference. So we found out that 27 is really that temperature threshold, and perhaps even 26 degrees Celsius. But what we see here is that at 25, we have this high degree of variation for our gametophytes and how they respond to that temperature treatment. And then at 27, they just completely lose their fluorescent intensity. And so when you look at the actual data, this is what this looks like. So 13, 21, and 23, not at all statistically significant. They're all basically the same. At 25, you have some gametophytes that do really well. You have some that really rapidly lose their chlorophyll fluorescence. And then you have some that are kind of in the middle. And then at 27, you see this very stark decrease right here. So we really were focusing on this 25 degree Celsius temperature because, again, this seems to have, like, it's very statistically significant, has a high degree of variation, regardless of population. So once we had that phenotypic data in the lab at the gametophyte life cycle stage, we wanted to compare it to biomass phenotypic data that had been collected from genetically similar adult sporophytes that had been outgrown on an open ocean farm here off the coast of Santa Barbara. So this data was collected in 2019, and we were able then to overlap what we saw. And I want everyone to focus just like what's going on in this purple circle, because these dots are actually those genotypes that we saw in a laboratory experiment perform extremely well, do very well under high heat stress. And they were the same genetic strain that also had the highest rate of biomass when outgrown in an open ocean farm. And I should also mention, when I say that they were grown under warmer than normal conditions, these juvenile sporophytes were outplanted on long lines from June to August, which is very late in the season for Southern California giant kelp. It's not their typical reproductive season, so we consider it to be warmer than average at that stage in their development. Okay, so moving on. Like I said, we were collecting IVIS fluorescent intensity data. At the same time, I was running confocal analysis. So this is what this looks like. The confocal is a 3D imaging system. It's not intervital, so these are fixed dead kelp cells. But as you can see here in this panel A, this is a whole kelp gametophyte. They look like little pom-poms. Each one of those little spindles is called a branch. Actually, this is a really good picture down here. This is a whole kelp branch, and these are individual kelp cells. The little dumbbell-like things are the chloroplasts, and in a healthy phenotype is when those chloroplasts are completely fringing that inner cell wall. So to compare IVIS to confocal, if you look at A2, we have this really happy, healthy, robust red-yellow splotch, which would correspond to a very high fluorescent intensity value. And then when you look at it under a microscope, the confocal microscope, this is what that would look like. When it's highly heat stressed, you see this stark decrease in fluorescent signal for the chloroplast or the chlorophyll. And that corresponds to the IVIS here. If you see like F2, this would be a very diminished fluorescent intensity value. So again, I didn't actually image the whole kelp gametophyte, I would just excise single branches. I would fix them in formaldehyde and seawater. And then I stained them with dyes that would actually adhere to nuclear DNA. So we have in yellow here is the nucleus and the microbiome. We did stain for the cell wall. It's not shown in these images simply because it kind of detracts from the overall picture. But what we see here on the left is the 13 degrees control treatment. So this is a healthy phenotype. We can see that the chloroplasts are evenly distributed within the cell wall. Each cell has a nucleus that is roughly in the center, and then the microbiome is very sparse and it lightly fringes the outer cell wall. The high heat stress phenotype without variation across the board looks like this. The microbiome is so incredibly proliferated. It's extremely bright. There's a complete loss of nuclear signal, so the nucleus goes away completely. And then the chloroplast will actually reshape and move to one side of the cell or the other. So that's the high heat stress phenotype. At 25 degrees Celsius, we have what is the intermediate heat stress phenotype. And again, we saw this without variation. So at 25 degrees Celsius, this is a single kelp branch. And you can see that some of them have nuclei. They have what appears to be healthy chloroplasts within those cell walls. And then some of the cells look completely unhealthy and they have that really high heat stress phenotype. So without variation, this is what we saw across all genotypes. And that leads us into part 2. So we wanted to perform a genome-wide association study, which is a very common follow-up to doing a phenotyping screen so that we can understand what genes, if any, might play a regulatory role in heat stress response. What we found from this, Maxim Kovalev in our lab actually ran this analysis, and he was able to find 3 significant single nucleotide polymorphisms, or SNPs. They're actually located on chromosome 3. So if you follow this right here, this dotted line means that anything above this is going to be highly, highly significantly correlated to our trait of interest. And in this case, we were looking only at those gametophytes treated at 25 degrees Celsius. And it just so happens that the 3 mutations or SNPs that we found are within the intronic regions, meaning they have the potential to change how those genes are expressed in the genome of a peroxidase, PRX gene, and an ATP binding cassette transporter gene. Why this is significant, in vascular plant systems or terrestrial crop systems, both of these genes are known to play roles in maintaining plant homeostasis under abiotic and biotic stress. So what's really cool is that the ATP binding cassette or the ABC transporters play a role in localizing the membranes of the chloroplasts, which, if you can remember, when they're under that high heat stress phenotype, what we saw with the confocal is that the chloroplasts actually like move and reshape themselves. So this is really illuminating and exciting. And then of course peroxidase or PRX play a huge role in just maintaining the stasis of the cell under high abiotic stress. So they detoxify and remove or actually maintain the reactive oxygen species levels inside the cell. So they play a really important role in just regulating that stress. So finding these was really fantastic, but again, it's just a start. There's more directions that we could take. I have a new graduate student in our lab, actually Karina Arzunian, is taking over my data set and doing RNA-seq, and so she'll be doing protein expression panels and some more exciting research with this data. So these are just kind of the first steps. But just to conclude, what we did with our research is we sought insights into the genetic basis of heat stress tolerance in giant kelp by integrating genomic analysis and fluorescence microscopy to identify key genetic markers. Hopefully we did that, which has hopefully some important implications for conservation of kelp forests, as well as breeding programs for enhanced resilience, in so much that we could potentially identify those individuals that are heat stress tolerant. We've just begun to do that, but of course we need to do functional validation and other steps just to confirm that what we are seeing is in fact real. But if this is what we think it is, then we could potentially start biobanking these strains or preserving them in some way for managed restoration efforts, of which there are many in California. So as Aaron mentioned earlier, we have the Kelp Forest Challenge, which Kelp Forest Alliance is very awesomely undertaking to restore 4 million hectares of kelp by 2040. The Nature Conservancy in California is playing a huge role in kelp restoration initiatives. My boss, Sergey Neustein, has developed Kelp Arc, which has a very robust seed bank for biodiversity initiatives, and several more. But yeah, I think that's the end of my talk. Thank you all so much. I really appreciate your time.
Lovely. Thank you so much, Madeline. A lot of very good information in there. Absolutely illuminating. As perhaps the general sort of million-dollar question, based on what you're preliminary finding, what are your hopes for using this in restoration? Is that idea that we can identify outplant more thermally tolerant kelp forests for the future plausible?
So what we found is that these strains, we did identity by descent analysis to determine how biodiverse, because each one of these strains is from different populations, and we wanted to determine, say, for example, you know, the worst happens and we have a bottleneck effect and a lot of our kelp forests get wiped out in the Southern California bight. Will there be any resilient strains that can bounce back? And if so, how biodiverse are they? Because that would matter in being able to, in population genetics standpoint, it would be really critical for those populations to be able to not only grow again, but also withstand other abiotic stressors. And we found out that unfortunately, the ones that do code for like high heat stress are also not genetically diverse. So one of the conclusions that we've made is that if we were to biobank and be like, oh, these strains have this potential for heat stress tolerance, they would need to be integressed potentially with the wild type or with other cultivars in order to maintain that standing or effective biodiversity that would be necessary for maintaining healthy populations. Yeah.
Okay, but certainly interesting first steps. Yeah, very promising. And we have a comment from Elizabeth that just says, go Trojans. So we'll give you that one. And then from George, we have, do you have any data on microbes in the control, intermediate, and high stress treatments? Or if they differed between genotypes, do you think the microbiome might play a role in that heat stress tolerance?
I definitely think the microbiome plays a big role. We didn't do any sort of analysis, but there are researchers in our lab who have been doing microbial work for kelp. And then also, if you check out Osborne et al. From 2022, she was one of our colleagues, and she did publish a Zeta diversity paper on the different microorganisms that are found in the kelp holobiont. But unfortunately, I'm not a source of knowledge for that.
Certainly another upcoming area. George, did you put your hand up and want to ask further? Are you happy? Silence indicates happy. Okay. Last question before we have to move on. A bit of a technical one from Chenille. How do you separate male and female gametophytes from Macrocystis?
Oh, absolutely. That's an easy one. So the females are really chunky. Their cells are much bigger than the males. So they're visibly dimorphic. When you grow them up after, say, like a few days to a week, you can begin to see that the female cells are, there's a lot less of them and they're much bigger. And then the males are much more like branching and filamentous, smaller.
Great. We'll have to, get some of this information in our next version of the Kelp Restoration Guidebook, because certainly some useful tips and tips in there. All right. Well, thank you very much, Madeline. We'll move on to the last talk, which will be me. I'll be chatting about a ecosystem monitoring and reporting guidebook that we've developed in collaboration with OceanWise. And the idea behind this is we have our kelp restoration database that we started a year or so ago, and trying to start providing guidelines and information on why and how we collect data for kelp forests. So essentially, we're just looking to standardize what we collect and then how we collect it. So when we have this big global database or other sorts of international collaborations, we can compare information across these projects, share information more easily, increase consistency, knowledge sharing. There's a bit more quality assurance when you know something's been collected to a certain standard, aids in tracking progress towards big global targets like the Kelp Forest Challenge, We can assess more sort of accurately the impact of our conservation interventions. We can just celebrate the success. We can talk about all the great things that happen when you restore a kelp forest, hopefully. And on the flip side of that, we can share our lessons learned from failure. So it's a bit of a dry topic, monitoring and reporting, but it underpins a lot of really important conservation outcomes, scientific outcomes, and just really, making the best use of all the good work and good effort that people are putting into these different conservation projects. So there are other sort of products that exist out there. Individual projects will often come up with their specific monitoring protocols based in a lab or a research group or a particular institution. We have big community science projects, citizen science projects like Reef Life Survey or PISCO, focusing on monitoring biodiversity. There's some sort of higher level UN global ocean observation systems. Different scientific papers have different methods. Various kind of biodiversity methods are starting to come out with biodiversity credits. And then we have the data platform, as I mentioned, where people are uploading information on their different restoration projects. But what we tried to do just a little bit differently with this one was have it be more holistic. So bringing together all different types of information you might be interested in recording or monitoring in a kelp forest ecosystem, beyond just the kelp itself or beyond the biodiversity, and designing it really with that sort of conservation focus in mind, and including a more holistic set, more social, more economic side of the question as well. And we also have this potential opportunity for uptake really with the Kelp Forest database and the projects that we're working with, and trying to create that as a more sort of centralized information system for how to monitor and report on these ecosystems. And lastly, we took a step-by-step approach. So a lot of the other information out there is perhaps a bit more high level, more generalized, but this has really step-by-step from 1 to 10 points of how you go out and count density or biodiversity or quantify primary production. And there's a few different methods for each service or function that you're trying to record. So there's some options depending on your capability. So I'll just provide a bit of an overview. We won't go through the whole guidebook in detail, but wanting to thank all the co-authors, Annie, Blanca, Scott, Dana, Nina, Maria, and Hannah for helping us put this together and really driving this into a comprehensive document that covers a lot of information. I'll send out a link along with all the other information from this webinar for the online version of it, but you can also find it on our knowledge platform on the cultforestalliance.com. And very high level, without getting too far into the weeds, the purpose of this talk is more just to get you aware of the document, know that it exists and it's there for use. We're not going to go really, really deep into the exact contents, but we have the basics, the principles of monitoring. So things like setting objectives, seasonality, your controls, your reference sites, how to set baselines, the different sort of costs of monitoring and how that factors into what you might want to choose. And then we also take a stab at creating key parameters. So things that we're trying to encourage different restoration, conservation, monitoring projects to record across all sites. And then at least we have some basic things to consider when we start to pull this data together. So we have kelp forest area, we have the kelp forest density, some measure of associated biodiversity, basic temperature, presence, absence, or abundance of herbivores or grazers, and then again the cost, just as we start to understand how to best spend our limited resources. It's a fairly easy thing to collect, but it's not often collected. And we also cover the bit of basics. So if someone's coming into this from a really sort of introductory standpoint, they can learn just a bit about what's a transect, what's a quadrat, what are the different tools, what are the logistics when you're creating your monitoring and reporting framework. And then in terms of what's covered, we have the kelp characteristics itself, so how to count and quantify those, regulating services like primary production, nutrient cycling, sedimentation, provisioning rates, so harvested kelp, secondary production, biodiversity, cultural services are touched in a bit of a lighter sort of fashion. There's a lot of information that's required to go into, say, how to measure somebody's cultural connection to a kelp forest. So a lot of that section points to existing resources and has a deeper dive available for people who are interested in quantifying those things. But we felt it important to include, to again encourage that sort of more social conservation-driven information collection for these restoration and conservation projects. And then your more standard biophysical stresses, temperature, salinity, herbivory. We try to encourage that everything's fair, so it's findable, accessible, interpretable, can be compared across projects, and reproducible, so it can be recreated based on the metadata, and then provide some examples of different local, regional, and international data repositories that you can host your information on, the Kelp Forest Alliance being one of them, but other international ones that I mentioned before, or perhaps your local, your regional conservation project might have their own database. So trying to provide information on where people can store their data so it gets the best use and the best view. If you're interested, after this, you can go on the kelpforestalliance.com and on the contact page, request an account. And from there, you can start to upload data from your own restoration projects, create your own organizational profiles, your personal profiles, browse through the people on the platform. There's about almost 600 people on the platform now. See what they're working on, see the different organizations they're tagged to, the different restoration projects, and start to use that as a way to contact people and access different areas of expertise and knowledge. If you want to learn more about green gravel or thermal tolerance, these sorts of people and information can start to become available on the platform. Jackie and Madeline, if you don't have accounts, I'll follow you down after this. So thank you for listening to that. I'll give you just sort of a brief overview as well on what the actual platform looks like. So this is the home page, and from here you can see all the different restoration projects that we have data for. So if you create the account, create a restoration project, it'll appear as a new dot on this map. You can also toggle by protected areas that exist for kelp forests, different organizations that we have mapped out within the network, and pledges that have been made to contribute to the Kelp Forest Challenge. But for now, let's just focus on different projects. And so you can zoom into a location. I'm going to be, hometown biased and go to Sydney and find some project details on this. And so this is what currently exists. This is a good example of the data that's been recorded in the past. And then we're trying to advance this a little bit by collecting a bit more comprehensive information. But you often have information on the restoration objective, site selection criteria, why it declined in the first place, the organization, the location, the species, motivations for conducting the research, different citations associated with it, and then the site observations themselves. So a bit of a summary of what was done, what lessons they learned, the project outcomes, and then how much area and what the percent sort of survival for this would have been. So ideally in the future, in addition to percent survival, we'll have things like fish biodiversity or invertebrate biodiversity or biomass or people employed or these additional variables and pieces of information that we're trying to encourage everyone to collect as part of their conservation projects. So there's a whole bit more that you're welcome to explore on the website. I'd encourage you to check it out if you're interested. But that is sort of the breadth and suite of the monitoring guidebook in a very short fashion. As I said, I will send out the link in the details. I think Scott might have popped through a link in the chat as well for anyone who's interested in reading it in more detail. And I can't ask myself a question. So does anybody have any questions about the monitoring guidebook, the platform, or any questions you might have missed for our previous two speakers, as we have about 5 minutes left with everyone? All right, we can have a last call going once. Let everybody get off a little bit early. All right. Well, that sounds lovely enough then. We will leave it there. We'll hopefully have a recording of this uploaded to our YouTube channel. You'll receive that follow-up email that I keep mentioning that'll have the link to the recording as well as the associated resources and bits of information. And we hope to see you at the next one. If you're not already part of the mailing list, you can also subscribe to that on the kelpforestalliance.com under the contact page, and you'll be sure to find out about following webinars. And this was very restoration focused, but we do talk about all sorts of topics. We had some art exhibits last time. We'll have some more sort of conservation fishery stuff next time, and really trying to illustrate and chat about all the different wonderful elements about kelp forest. So thank you all very much for joining us. A big thank you to Madeline and Jackie for their wonderful talks. And we'll hopefully see you all again soon.